Now that we know a little bit about how microorganisms grow, we can talk a little bit about controlling their growth. We'll start with some terminology. Sterilization is the removal or destruction of all microbes, including viruses and endospores, from a substance. Something that has been sterilized is said to be sterile. There is nothing on its surface. Commercial sterilization is used on food. It does not kill all hyperthermic microbes, but none of these are pathogenic, and since most canned food is stored at room temperature, these organisms won't grow at room temperature. Aseptic means an environment is free from pathogens. Aseptic technique is a technique that prevents pathogens from contaminating the area. When we destroy or inhibit microbes on an inanimate object, we are said to have disinfected it. The focus is always on pathogenic organisms. A disinfectant is a physical or chemical agent that's used on a surface to disinfect the surface. Antisepsis is the inhibiting or destroying of microbes in a tissue. Again, the focus is on pathogenic microorganisms. An antiseptic is a chemical agent that's used on tissues to inhibit or destroy microbes. Some antiseptics and disinfectants are the same chemical. Sometimes it's the same chemical at a different concentration. But typically antiseptics have to be a little milder than disinfectants because you're dealing with human tissue. When someone is septic or when there is sepsis, that means that microorganisms are growing in the blood or the tissues, or their toxins are present in the blood or tissues. When you wash your hands, you are degerming them. You are removing microbes from the surface of your hands by scrubbing. Sanitization is the mechanical removal of microbes to some acceptable health standard. Pathogens may still be present, but they're present in very small numbers. Sanitization is used in restaurants and public toilets. Pasteurization is used to remove pathogens or unwanted organisms from food typically. It helps prevent spoilage, it prevents the spread of pathogens in the food, and in some instances it's been used in wine and beer to stop the fermentation process. When a word ends in stasis or static, it means that it inhibits the microbial metabolism and growth of an organism, but it does not kill it. If the word ends in "-side" or "-sidal", then that destroys or permanently inactivates a group of organisms. So a viricide inactivates viruses. Bactericides kill bacteria. Fungicides kill fungi, and germicides kill germs or pathogens, and only pathogens, not all microorganisms. Microbial death is when an organism permanently loses its reproductive capability, even when it's placed under optimal growth conditions. A technique for evaluating the efficacy of antimicrobial agents is to look at the microbial death rate. Death in microorganisms is a logarithmic function, just like growth was. The most susceptible organisms in the population will be the first to die, the oldest, the weakest, the ones that are most susceptible to the agent being used. Those that are the most resistant will die last. This is why it is important to use and apply microbial agents the full recommended time. Otherwise, you're going to leave behind the resistant organisms and you will select for resistant strains. There are basically four ways antimicrobial agents work. They can damage the cell wall. Lysozyme and penicillin work this way. They can damage the cell membrane. This is how most surfactants work. They can affect protein or nucleic acid synthesis. Chloramphenicol and radiation employ this method. Or they may alter protein function by denaturing the proteins. Since all enzymes are proteins and enzymes control all metabolic pathways, this pretty effectively shuts down metabolism in the cell. Heat, alcohol, and changes in pH can denature proteins. When we're trying to evaluate the effectiveness of antimicrobial methods, there are some things we need to take into consideration. First of all, what is it we're trying to disinfect? We have to choose a method that's appropriate for the material being used. Plastics can't be heated, for example. How clean does it have to be, and what can the material endure? Anything that's going to penetrate the skin has to be sterile, but things that are going to be used on the skin surface only have to be disinfected. What kind of microorganism are we trying to get rid of? Low-level disinfectants work well on vegetative cells and envelope viruses. Medium level kill most fungi, most viruses. High level disinfectants are needed if we're trying to get rid of mycobacterium, endospores, or prions. And the third thing we have to consider are the environmental conditions. Higher temperature and lower pH will enhance the effects of any antimicrobial substance. If there is a lot of organic material present, that may hamper the effects of an antimicrobial agent. Some can work in the presence of organic materials, others cannot. We also have to know the bioburden we're dealing with. In other words, how many microorganisms are there and what type of microorganism are they? Are they high level and there's a lot of them? Are they low level and there are a few of them? This bioburden influences how long and how much antimicrobial agent we need. Here's an example from most susceptible to most resistant of microorganisms. Most things that have an exposed cell membrane are pretty easy to kill. As we get up here to prions and bacterial endospores and mycobacteria, which has mycolic acid in its cell wall, we have more resistant things, things that are harder to kill. It would be nice if we could find one antimicrobial agent that would do it all. The ideal antimicrobial agent would be inexpensive, fast-acting, be able to be stored pretty much indefinitely, would control the growth and reproduction of every type of microbe and be completely harmless to humans, animals, and objects. Obviously, this antimicrobial agent does not exist. You will see a number of antimicrobial agents selected, but every agent has its limitations and its disadvantages. That's why you see such a multitude of agents used in various situations. Biosafety levels were set by the CDC as guidelines for microbiology labs. This is what microbiology labs can handle safely. Biosafety Level 1 or BSL-1 labs can handle microbes that are not known to cause disease in healthy humans. They can use minimal precautions, hand washing and cleaning of surfaces. BSL-2 labs are moderately hazardous. They can handle infectious agents like hepatitis B and MRSA. They have limited access. All sharps are disposed of in a specific way, and they limit the production of aerosols. BSL-3 labs have all manipulations done in biological safety cabinets that have high efficiency particulate air filters. They also have a double door entry. And BSL-4 labs handle the most hazardous materials. These are the most secure labs. They handle dangerous or exotic microbes. People who work in these labs are the ones that dress up in the spacesuits. These labs are completely isolated from other areas of buildings. They don't share the same air source. They have separate entryways. They're incredibly secure. We can control microorganisms either using physical means or chemical means. We'll look at physical means first. These will be heat, cold, drying, filtration, osmotic pressure, and radiation. Heat is an older and more common method for controlling microbial growth. Heat will denature proteins, it interferes with the cell wall and cell membrane function, and it can disrupt nucleic acids. Microbial susceptibility to heat will vary among organisms. So we have something called the thermal death point. This is the lowest temperature that kills all cells of a particular organism in a broth in 10 minutes. The thermal death time is the time it takes to completely sterilize a particular volume of liquid at a set temperature. Again, this is for a particular organism. The decimal reduction time is the time required to destroy 90% of microbes in a sample. The decimal reduction time can be used to help determine how long an organism needs to be exposed to heat or any other physical or chemical means of disinfection in order to kill the entire population. So microbial death is a logarithmic function, so we have the powers of 10 over here. In 1 minute, 90% die. Every minute, 90% die. Eventually, we'll get down to where we only have 1 or 2 cells, and it will be impossible to kill only half a cell. That last period of time will be the maximum time needed to kill everything in the population. Moist heat denatures proteins and destroys cell membranes. It's more effective than dry heat because it penetrates better and steam is a better heat conductor. One form of moist heat is boiling water. This will kill vegetative cells of bacteria and fungi, trophozoites of protozoa, and most viruses within 10 minutes if you're at sea level. Everything but endospores will be destroyed by boiling for 30 minutes. Endospores can survive boiling for 20 hours, so boiling is not really a way to get rid of endospores. Boiling is very effective for food and utensils and bedding, but the biggest problem is when you take it out, it's recontaminated immediately. Another form of moist heat is autoclaving. This is steam under pressure. When you put steam under pressure, it increases the temperature of the water. An autoclave is basically a pressure chamber with pipes that remove air and introduce steam, and we have temperature and pressure gauges to monitor the pressure and the temperature inside the chamber. The standard for autoclaving is 121 degrees centigrade at 15 pounds per square inch pressure. The time we use depends upon the bioburden. If things are small and relatively clean, 15 minutes is an effective time. If things are larger or we have more contaminated loads, then it requires 30 minutes. For example, the trash we discard in lab requires 30 minutes of autoclaving because it has a large bioburden. Here you see a basic autoclave, the interior chamber. We have a way to let air out. We let steam in. Steam gets into the chamber. As the steam comes into the chamber, the air is forced out. When we have enough steam in there, we close off all the valves, continue adding steam to build the pressure. Once the pressure is there, we close off the steam, allow the steam to do its job for the appropriate amount of time, and then release the steam. Steam has to penetrate all of the items in the autoclave, so only special wrapping materials can be used, and the package can be only a certain size. Also, when we load the autoclave, we have to make sure that there's space between all of the packages so that steam can circulate freely. In order to make sure the autoclave is being effective, there are quality control measures done. Every load has tape in it. All of the items are taped with this tape. The tape will change color if it's exposed to steam. This does not mean the appropriate temperature was met, but it does mean that steam got to the tape. In order to check the efficiency we use Bacillus styrothermophilus. The endospores of this organism are the most heat-resistant endospores. Spores from this organism are placed in the center of one of the packages in a load. After autoclaving, the spores are removed and placed in growth medium. If they grow, we know that the autoclaving was not effective. If they don't grow, then the autoclaving was effective because these spores were killed. Pasteurization is another form of moist heat. It's used to destroy certain microorganisms based on their heat sensitivity. It's used primarily for pathogens in milk, but juices, wines, and beers are also pasteurized. When we pasteurize milk, we're targeting Brucella melitensis, Mycobacterium bovis, and E. coli. Brucella causes undulant fever, Mycobacterium causes tuberculosis, and E. coli can cause rather severe diarrhea. Pasteurization is a disinfection technique. Not all microorganisms are killed, only the targeted ones. Pasteurization time will vary with the product. When you're doing the batch method for milk, you want to heat the milk to 63 degrees centigrade and hold it at that temperature for 30 minutes. That will not change the flavor of the milk and will effectively destroy the organisms that we're targeting. Most commercial producers of pasteurized milk use flash pasteurization. Milk is sent in thin streams across a surface where it's heated to 72 degrees centigrade for 15 seconds and then very quickly cooled. This does not change the flavor of the milk and is still a disinfection process. Ultra-high temperature, or UHT pasteurization, is flash heating. The milk is heated to 140 degrees centigrade for 1 to 3 seconds. This does sterilize the milk, but it may affect the taste. UHT is used a lot in Europe, and if you've ever been to Europe and tasted the milk, you may have thought it tasted a little funny. In this country, we tend to sell this in juice box type things, and it's flavored with either chocolate or strawberry flavoring to cover the taste. Dry heat is used to sterilize powders and oils and other things that can't take moisture or pressure well. Dry heat also denatures proteins, but you require higher temperatures and longer times. 121 degrees centigrade dry heat would require 16 hours to sterilize something. Usually, we use 171 degrees centigrade at 1 hour or 160 degrees centigrade for 2 hours. Dry heat is not suitable for rubber, paper, or plastic. The other form of dry heat is complete incineration. This is very effective. We use this when we sterilize our loops in lab. We can burn contaminated medical material. We can burn diseased carcasses. In some cultures, burning the dead is how you get rid of the body, and that's a very safe way to do things, particularly if the person died of anything infectious. However, incineration is very destructive. Ashes are sterile, but that's all you're going to have left is ashes. The other extreme is cold. Refrigeration is temperatures between 0 degrees centigrade and 7 degrees centigrade. This will decrease microbial growth except for those organisms that are psychrotrophs. Pathogens are generally affected because they're mesotrophs. Listeria is one organism that's pathogenic that can reproduce at 4 degrees centigrade. It's found in cheeses, dairy products, and delicatessen meats. Yersinia pestis can multiply in refrigerated blood products. Yersinia pestis is the cause of bubonic plague. Freezing is another application of cold. This is temperatures below 0 degrees centigrade. If something is frozen slowly, the ice crystallizes, and this pokes little holes in the cell membrane. So slow freezing usually kills most things. However, many bacterial cells, bacterial endospores, and viruses will survive freezing. Once they thaw out, they'll start to grow again. Drying or desiccation has been used as a food preserving technique for years. It inhibits microbial growth by removing water for metabolism. This will inhibit most pathogens. However, dried materials are susceptible to molds because they can grow in that drier atmosphere. Lyophilization is freeze drying. Here we use two techniques to preserve microorganisms, and this is actually a preserving technique. First, you quick freeze them by taking them very quickly to sub-zero temperatures, then you use a vacuum to remove the water. Filtration is the passage of fluid, either a liquid or a gas, through a sieve that's designed to trap particles. You frequently use a vacuum to pull the fluid through the filter. The pore size of the filter determines what is trapped and what gets through. This is used to sterilize all kinds of heat-sensitive materials. Membrane filters can trap viruses and even some large molecules, so we have filters with incredibly small pores. The medical applications are the filtration of high-protein nutrient solutions and the use of HEPA filters. These filters are used in biological safety cabinets. They may be used to sterilize the air in operating rooms. The rooms of patients with airborne diseases like tuberculosis will filter that air before it's released to the general population. It's also used to filter the air going into rooms of immunocompromised patients to make sure no microorganisms get in that way. Osmotic pressure is the use of salt or sugar to draw water out of a cell. This is an ancient form of food preservation. The high osmotic pressure prevents microbial growth because the cells lose water via osmosis. Honey, jerky, jams, jellies, salted fish, and some pickles all are preserved based on osmotic pressure. Molds can still grow because they're not as susceptible to low osmotic pressure. There are two basic forms of radiation. Ionizing radiation is electron beams, gamma rays, and some X-rays. These eject electrons from molecules, causing oxidation, and does considerable damage to DNA. Electron beams do not penetrate well, so they don't go through organic material, but they can be used to sterilize the surface of meats, spices, and microbiological plasticware, dental, and medical supplies. Those things that are prepackaged have usually been sterilized using radiation in some form. Gamma rays have been used on food. Irradiating the surface of the food removes microorganisms and will increase the shelf life of produce by several days. Many people are very concerned about eating irradiated food. Non-ionizing radiation is ultraviolet light, visible light, and infrared radiation. UV light will cause thymine dimers in DNA. Ultraviolet light does not penetrate well, but it can be used to disinfect air and to maintain the cleanliness of surfaces. In addition to physical methods, there are chemical methods that can be used for disinfection and antisepsis. The chemicals may be solids, liquids, or gases. They may be disinfectants, antiseptics, or even sterilants, things that are capable of sterilizing material. Chemicals may come in aqueous or water-based solution or in tinctures, that is, dissolved in alcohol. The categories of chemicals used for disinfection and antisepsis are phenolic compounds, alcohols, halogens, oxidizing agents, surfactants, heavy metals, aldehydes, gaseous agents, and enzymes. Phenolic compounds are all based on phenol. Phenol was first used by Lister in antiseptic surgery. The phenol he used was carbolic acid, and carbolic acid is poisonous and very hard on the hands. For many years, phenol has been the standard to which all other disinfectants was held. Phenolic compounds are chemically modified phenol molecules. Phenolic compounds denature proteins and disrupt cell membranes. Cresols are one form of phenolic compounds. Cresols are what are found in Lysol. Bisphenols are two phenol rings held together chemically. Hexachlorophene and triclosan are both bisphenols. These are both skin antiseptics, and they both have sort of a time release effect. That is, they tend to linger after you have washed them off. Visahex uses hexachlorophene, and triclosan has been incorporated into pretty much everything. You can get antimicrobial trash bags, antimicrobial diapers, antimicrobial soap, antimicrobial plasticware. Just read it. Somewhere on it, it will say triclosan has been incorporated into it. Natural oils like pine oil and oil of clove are also phenolic compounds. Phenolics are effective even when there is a large amount of organic material present. They're widely used in healthcare settings, labs, and homes. They still may irritate the skin, and they have that medicine smell that many people find unpleasant. Alcohols dissolve membranes. Isopropanol and ethanol are the two most common alcohols used for antiseptics. Tinctures are usually more effective than aqueous solutions of the same chemical, so many other disinfectant chemicals may be dissolved in alcohol, and the combination will make both of them a little more effective. 70% alcohol is actually the most effective. This is a case where 100% is not the way to go. Because alcohols evaporate, it diminishes the contact time alcohol has with a surface, making it not the best of disinfectants. It is used to damp dust in surgery. Dust in surgery is not a good thing because that dust may be carrying microorganisms. By using an alcohol, you control the dust, plus it does give a nice shine to stainless steel. Alcohol is also used for degerming the skin prior to injections. That alcohol pledget that you scrub really hard with, and that helps dissolve the skin oils where most of the bacteria are found. And so you're mechanically removing material, not really disinfecting the skin. Halogens are those chemicals, iodine, chlorine, bromine, and fluorine. They denature proteins. Iodine is usually seen as tincture of iodine and is used as a skin antiseptic. Iodophores are slow-release iodine-containing organic molecules. These are also used as skin antiseptics. Betadine is a very common iodophore. Chlorine is a sanitizer. It's used in water to chlorinate water. Also, chlorine is what is in household bleach, which is an effective household sanitizer. Bromine is used in hot tubs because it evaporates more slowly at high temperatures, and fluorine is used in water and toothpastes. Fluorine will help reduce dental caries. Oxidizing agents are things like peroxides, ozone, and peracetic acid. These release oxygen radicals, and oxygen radicals will destroy anaerobic bacteria. Hydrogen peroxide is used commonly in the household. Many people erroneously pour it on open wounds to sanitize open wounds. Human tissue has catalase in it, so that peroxide is immediately broken down to water and oxygen and has no killing power left at that point. It is good if you want to bubble something out. The oxygen bubbles will help gently cleanse something, but a different disinfectant or antiseptic is needed after using peroxide. Hot peroxide is used by food processors to sterilize packaging, like drink boxes. Ozone is used to disinfect water. It's actually more effective than chlorine, but it's more expensive and it's harder to maintain an appropriate level of ozone in the water. Peracetic acid is actually a sporicide. This can be used to sterilize equipment. It's not adversely affected if there are large amounts of organic material present, and it doesn't leave a toxic residue. Peracetic acid is used to clean equipment in both the food and medical industries. Surfactants are wetting agents. They reduce the surface tension of liquids. Adding surfactants to almost any disinfectant gives you a sheeting capability if it's in an aqueous solution. That means the disinfectant will be applied uniformly to all parts of the surface. Soaps are the most common surfactants. Soaps have a hydrophobic end and a hydrophilic end. The hydrophobic end will interact with oils. The hydrophilic end will interact with water. This allows soaps to remove oils and dirt and bacteria that may be in the oils and allow it to be washed away by water. Soaps are primarily for mechanical removal. They really don't have any killing capability. Detergents are for the most part cationic agents. These will disrupt cell membranes. Detergents are typically more soluble than soaps. Quaternary ammonium salts are probably the most common detergent used medically. It's colorless, tasteless, odorless, and totally harmless to humans. If you have a foaming mouthwash, it probably has a quaternary ammonium salt in it. Benzalkonium chloride or Zephyrin is probably the most common of the quaternary ammonium salts, and you can find this in Cepacol mouthwash. Heavy metals are things like arsenic, zinc, mercury, silver, and copper. These all denature proteins. They are toxic to humans, and many people are allergic to some of these metals. They're also easily inactivated by organic material. Thimerosal is a preservative. It has mercury in it. The MER in there tells you it has mercury in it. It used to be used in contact lens solutions, but people started having allergic reactions and they stopped using it. It is being withdrawn from vaccines, but it is still present in a few adult vaccines. Merthiolate is a skin antiseptic. When I was growing up, you had your choice of two things to go on your cut, either iodine or merthiolate, and we always wanted the merthiolate because it was in an aqueous solution and it didn't burn. Copper can be used to kill algae. It's used around swimming pools and koi ponds to help keep algae from overgrowing. Zinc, copper, and mercury have been incorporated into paints to help prevent mildew. Aldehydes also denature proteins. One aldehyde is glutaraldehyde, known under its brand name of Sidex. This is used in surgery to disinfect endoscopes. Endoscopes, because of the cameras involved and the tubing, can't be autoclaved and are pretty sensitive to most chemicals. Soaking the endoscope for 10 minutes in Sidex will disinfect it. If you soak it for 10 hours, it will actually sterilize it. Another aldehyde is formaldehyde. A 37% solution of formaldehyde is known as formalin. Formalin is used in embalming, and it's been used to disinfect hospital rooms, instruments, and machines. Formaldehyde is typically how they disinfect dialysis machines. You have to be sure that all the formaldehyde is removed from any machine because it will irritate mucous membranes. Formaldehyde is also carcinogenic. Gaseous agents can be used in closed chambers to sterilize large equipment, plastics, electronics, things that can't be sterilized with heat or water, or things that may just be too big to sterilize any other way. Most of these gases leave a toxic residue on the surface of whatever they sterilize, so there has to be an aeration period so the gas can dissipate. Most of the gases are also carcinogenic and explosive if not mixed with other agents like nitrogen or carbon dioxide. All of the gaseous agents act by denaturing proteins. Ethylene oxide is the gas used in the chemiclave or the gas autoclave. Most hospitals have at least one gas autoclave to sterilize delicate equipment that cannot be sterilized using steam. Beta-propiolactone is frequently pumped into warehouses that hold nuts or spices to disinfect those items. And chlorine dioxide is actually a halogen, but it's in its gaseous form. It can be used to disinfect drinking water, wastewater, food processing equipment, and medical waste. Lysozyme is a naturally occurring enzyme found in tears and other secretions. Recently, it's been used to remove microbes from cheese and wine. Some vintners use sulfides to stop the fermenting process in wine. This leaves behind a residue of sulfur. Some people who drink the wine are allergic to sulfur and get headaches. Using lysozyme overcomes this problem. Preenzyme is a new enzyme that destroys prions. In 2006, it was approved by the European Union to treat surgical instruments that had been used in brain surgeries where people had prion-type diseases. This means that it's been approved for use in Europe, but it's not been approved by the FDA, so it's not in use in this country. This is the first certified non-caustic chemical that can treat prions. How do we evaluate disinfectants and antiseptics? Well, the usual considerations, the higher the concentration, the fresher the solution, and the longer we expose it, typically makes a disinfectant or an antiseptic more effective. But if we really want to test it, we can use the phenol coefficient. For years, this has been the standard. You compare the efficiency of the disinfectant to phenol. A phenol coefficient of 1 would mean it's just as effective as phenol. A phenol coefficient of 2 would mean it's twice as effective, and a phenol coefficient of 0.5 would mean it's half as effective. The use dilution test uses 3 organisms, Pseudomonas aeruginosa, Salmonella enterica, and Staph aureus. These are 3 very resistant organisms. You use these organisms to coat cylinders that you dip in the disinfectant you're testing, and then after rinsing the disinfectant off, you place the cylinders in broth for 48 hours. The most effective disinfectant is the one that prevents growth at the highest dilution. This has now become the current standard in the United States. The Kelsey-Sykes capacity test is the standard alternative that's been approved by the European Union. So this is the one that's used in Europe. Here you add a suspension of organisms to a suitable concentration of the chemical you want to test. At predetermined times, every minute, every 10 minutes, whatever it might be, you remove a sample of the mixture to a growth medium that contains a disinfectant inactivator. You want to make sure that the disinfectant doesn't travel with the organism. You then incubate the tubes for 48 hours and you look for no growth. You look for no turbidity in the tube. That would mean that the disinfectant killed or prevented the growth of the microorganism. The in-use dilution test is probably the most effective test, however. Here you take swabs from the actual equipment before and after the disinfection procedure. Then you use the swabs to inoculate media, incubate them, and look for growth. People are always talking about resistant microorganisms. So how do they develop? How do we get them? In this country, we are absolutely preoccupied with antisepsis and disinfection. We have more disinfectants on the market. We have all kinds of things that are antibacterial, things that don't really need to be antibacterial. There is little evidence that any of this has added anything to the health of individuals, and yet we continue to do it. This means that a lot of these items may be misused, and that is a problem. Remember, we're going to kill the most susceptible organisms first. If we don't use enough, if we don't use it correctly, then the resistant organisms are left behind. Soon the environment will contain only the resistant strains. That's not a good thing. As soon as you have a population of only resistant organisms, then you have a resistant population, and that particular disinfectant or antiseptic will no longer work on it.